Hi, Gen Bio. Dr. Fry here, and today we are going to be talking about plant growth regulators. So, we're going to look at a few concepts that enable the plant to exchange gases with the environment. So, obviously, they need to have CO2 and they need to release oxygen as a part of photosynthesis. And this happens, as we'll see, through small pores called stomata. And those stomata are regulated by a type of cell called a guard cell. So we're going to look at that as well as the concept of apical dominance. Apical dominance refers to the fact that the main stem of a plant has a dominant or preferential growth to its lateral branches. Okay, so we're going to look at those concepts today. But we need a little bit of background on leaves first, just so you know what they look like and under the microscope what you should expect to see when we take a closer look at a leaf. So the leaf is a part of the plant that is responsible for making food through photosynthesis. There's lots of chloroplasts, and the leaf is designed to harvest a lot of energy from sunlight. Okay, so let's look at some external anatomy of the leaf. So what we often refer to the leaf is referred to as the leaf is technically called the blade. Okay, so this is the blade, and this is a diagram, and this is obviously a actual blade here, specimen. Okay, the blade is covered by two layers of epidermis, one on the top of the leaf and one on the bottom of the leaf. And then it is additionally covered by this waxy coating called a cuticle. Okay, and this helps with water retention and it keeps the leaf from drying out. Okay, so that's the blade, and there are veins running through it, as we looked at during our last lab. Depending on whether or not the plant is a monocot or a dicot, those leaf venations may be parallel or branching. Okay, so those are the veins. You can see they're going to hold the xylem and the phloem. And then connected to the blade is a small leaf stem called a petiole. Okay, so shown here and then over here as well. Okay, and then at the base of the petiole, you might find little leaflets, little tiny things that look kind of like a leaf. Those are called stipules. Okay, down here, there's a nice example here. It's very small at the base of the petiole. Okay, and then of course, there's the main stem. And sometimes at the branch points of different petioles, you might see axillary buds. So buds are where there is growth taking place, another branch coming off, and you might see those in the crooks between two different petioles. So those, if they're in the wedge in between, then those are called axillary buds. Okay, versus the apical bud, which would be the dominant one. Okay, so let's move to our next slide and take a look at some internal leaf anatomy. So as I mentioned, on the outside of the leaf, this would be the top outside surface, and as well as on the underside of the leaf, there is a single layer of epidermis. So here, as well as here. And then within that epidermis, there are several pores called stoma, or plural stomata. Okay, and those pores are created by two specific types of cells, guard cells, that flank the pore. So they kind of like form a hole in the middle, with my hands being the guard cells. And they can open and close the pore based on the turgidity of the guard cell. So how much water, how much pressure is inside the guard cell will change the shape of the pore. Okay, and then so we will get to that in further detail in just a minute, but I'll just mention that you can see here the vein with the xylem and the phloem running through the leaf, and then there's some mesophyll layers inside the two layers of epidermis. I'll also mention that you are going to, when you do the experiment in lab this week, you're going to find a greater abundance of stomata on the underside of the leaf. You're going to see more present there. There are some on the top layer, but most of them will be on the underside. So when you're doing the lab activity this week, you want to look at the epidermis on the underside of the leaf specifically. Okay, so this picture is showing you a stoma, the pore, that I said is flanked by two guard cells. Here's one, and here is the other. And they have a unique morphology to their cell walls. So they have a thicker inside cell wall, as well as some supportive structures, which allow for the bowing of the guard cells and the opening of that pore when water enters the guard cell. So as water moves in, it's going to create this bowed appearance to the guard cell, which then in turn causes the opening of the stomata, okay, of the stoma. So with this, we obviously need to do this. The cell needs to open stomata in order to exchange gases with the environment. So you know that when photosynthesis is taking place, carbon dioxide is used and oxygen is released. So let's move to our next slide to explore that and what happens with that a little bit further. So if you need a little refresher, the equation for photosynthesis is right here. 6 carbon dioxide and 6 water molecules with access to light energy from the sun can be converted to glucose and 6 oxygen, right? So with the light energy coming from the sun, photosynthesis is taking place at higher rates. And this causes a decrease in carbon dioxide in our guard cells. Okay, so there's a lower concentration of carbon dioxide when it is conducting photosynthesis. Okay. And this is going to turn on a mechanism in the guard cell which allows for potassium ions to move into the guard cell from surrounding cells. So when you have a decrease in carbon dioxide in that guard cell, it's going to turn on open channels that allow for potassium to move into the guard cell. And then other ions will accompany potassium. You'll notice that potassium is a monovalent cation, and it will be accompanied often by the movement of a monovalent anion, usually chloride. So those will both move into the guard cell. And because of what we know about osmosis, water is going to follow the movement of ions to even out the concentration gradient. So water is going to flow into the guard cell, and that is what's going to increase the turgor pressure. So the increased pressure through an increase in water inside the guard cell is what's going to allow the stoma to open. Okay? So with photosynthesis, with an increase in light, with an increase of potassium inside the guard cell, these are all factors which will lead to the opening of stomata. Okay, so just to represent this a different way, I kind of wrote it out here in word outline form. Factors that cause stomata to open include light, decreased CO2 in the guard cells, potassium moving into the guard cells, and to a lesser extent, there have been studies that indicate that some other monovalent cations, such as sodium, can also increase the likelihood of stomata opening. Water will follow. So all these factors cause stomata to open. Additionally, an increase in temperature, as well as the plant's natural circadian cycle, which is similar to our own. Different genes are turned on with the new day, with the light coming out, with our normal circadian rhythms. This will also open stomata. Okay. On the other hand, the factors that are linked to closing of stomata are kind of the opposite end of the spectrum. So an increase in CO2 in the guard cells. If there's a lot of carbon dioxide in the guard cells, then we're not going to have the stomata open because we don't need to increase that further. Wilting of the plant due to a water deficit will also cause closing of the stomata. And the way that this happens, the mechanism of action, is through release of a plant hormone called abscisic acid. Abscisic acid triggers calcium entrance into the cell, into the guard cell. And this results in lower potassium inside the guard cell. So this is through closing the channels that allow for potassium to enter, and also causing an increase of movement of potassium outside, towards the outside of the guard cell. So water will follow that movement of potassium. So you're getting less potassium in the cell, in the guard cell. Water is going to follow by moving out. That's going to decrease the trigger pressure inside the guard cell and close the stomata. Okay, so we can sort of hijack that natural mechanism of action by applying calcium to the cells or to the surrounding solution when we do our experiment in lab. So that is exactly what we're going to do. We're going to apply different solutions to the epidermis of a leaf, and we are going to observe the effect on the stomata. So for our experiment, we are going to be putting epidermal strips that we've removed from the underside of a leaf into different solutions. So the solutions we'll be using are potassium chloride in a lighted environment, potassium chloride in a dark environment, sodium chloride, calcium chloride, and MOPS. MOPS is just our buffer, so it's going to act as a control for us. And then the other solutions involve potassium, which is a monovalent cation, and chloride, which is a monovalent anion, and comparing those in light and dark, as well as looking at sodium chloride. Sodium is also a monovalent cation, and calcium chloride. Calcium is a divalent cation, and we had talked about the effects of calcium on closing of the stomata. So what we should expect to see is that potassium, because of what we know about guard cell movement, should cause opening of stomata. Now, we also know that light is a factor that influences opening of the stomata, and dark would normally act to close the stomata, to retain water in the plant, keeping that stomata closed so it doesn't lose water vapor, to retain water in the plant when the plant cannot be undergoing photosynthesis because there's no light, right? So we're going to compare those to other solutions that have different ions. Sodium, as I said, is a monovalent cation similar to potassium. So we'll see if that has as much of an effect or a similar effect at all to potassium. And then calcium, as we had mentioned in our last slide, should act to close the stomata. And then I'll just point out, and I want you to think about a little bit, the fact that chloride is involved in each of these solutions. It will be present in all of the ones that we test. So give a little thought to what effect chloride could be having as we analyze our results. For instance, if we see different results in the potassium versus, say, the calcium chloride solution, what action is chloride really taking on here? Is it having an effect on its own, or does it seem to be sort of a passive agent here? So I'd like you to keep that in mind as we do our experiment. And then what our job is to, after the epidermal strips have sat for 40 minutes in the solutions, the various solutions, we are going to analyze the size of the pore that we can see here. So all of these little jigsaw puzzle type shaped cells, those are parenchyma cells in the leaf. And then the guard cells are these here, 1 and 2. that surround and regulate the opening of the pore. So we're going to take a look then to see how many stomata are wide open, open, or closed when placed in the various solutions. And we're going to compile that data and take a look at that in more depth. Now let's move on. We're also going to set up a second experiment this week, which looks at apical dominance. Now you won't be able to analyze this one in lab this week because we're going to let it sit for 2 weeks. And then we'll take a look at the final results after it's time to grow. So the concept that we're looking at is apical dominance, which is the phenomenon where plants' main stem here has a preferential growth over the lateral branches, so the branches coming out to the sides. Okay. And the way that this works is through the action of a group of hormones called auxins. These are plant growth hormones. The one that we are going to be using as an auxin in lab this week is naphthalene acetic acid, and we're just going to call it NAA for now, naphthalene acetic acid. And so these auxins are produced in the apical meristem, so at the very tip of the main stem. And so they're produced at the tip, at the apex, and travel down towards the base of the stem. And as they move down, they are able to promote cell division of the apex and suppress growth of the lateral branches. And this works through an activation of another hormone called strigolactone. And strigolactone suppresses the lateral growth of the branches. So if we were to remove the apex of our plant, we should expect that this hormone could not act to suppress lateral growth because we have removed it. It's produced in the apex. So we clip off the tip, then it's not being produced and it's not suppressing lateral growth. So what we should see then is increase in lateral growth with the removal of the auxin in the apex. Okay, so that's what we're going to do in lab. We're going to set up a number of conditions where we have a control, and then we're going to have a stem removed and coated. You're going to coat the cut end with lanolin. Lanolin is a protective agent. And sometimes we're going to include with that lanolin various concentrations of our auxin, naphthalene acetic acid. So with the increase in auxin, then we should see, to some degree, an increase in suppressed lateral growth or a decrease in lateral growth. With no auxin, just lanolin, what we should see is that then that lateral growth is free to continue without any suppression. Okay, so we will set that up this week. The plants we're going to use are bean plants, Visha fava, and this is the main stem. So we'll cut off the apical stem here and examine the growth of our axillary buds. So you're going to measure the growth that takes place when we cut off the apical stem and then either apply an artificial auxin in place of the natural one, or just a control. We'll apply just a lanolin coating. Okay, and then in 2 weeks we're gonna analyze the results. Okay, so for this week's lab, you're gonna start by setting up the guard cell experiment by removing the epidermal strips. You're just gonna pull off the epidermal strip, and I'll demonstrate this in class. And place those strips in different solutions that include different combinations of either potassium chloride, sodium chloride, calcium chloride, or our buffer MOPS. And then we're going to let that sit for 40 minutes. And then we're going to tabulate the results at the end of that 40 minutes. In the downtime, what you're going to do is you're going to set up your apical dominance experiment. So you're going to use plants that we will place in the greenhouse after you have cut the apical stem off and measured the axillary buds. Okay, we'll check that in 2 weeks. And then I also want to mention that this experiment on the stomata is going to be the experiment that we use to write your lab paper. So you're going to compile one large lab paper for the end of the semester that has all of the component parts that we've worked through so far. So that we haven't worked through the abstract yet, but that is just sort of a summary. You're going to include the most important sentence or two from each of the other sections. Okay, so you're going to have one or two important lines from the introduction, basic summary, one or two important lines from how you conducted the experiment, one or two important lines from the results, same with the discussion. Okay, that's the abstract. Then you're also going to write the introduction for this experiment, materials and methods for the stomata, results, which we'll compile, and a discussion. Okay, so this is going to be your final lab paper. All right, so that's it for today. We will see you in lab this week, and I hope you guys are all enjoying the beautiful weather. Have a great day. Bye-bye.